So it's a pleasure to be here and see so many familiar faces and new faces, and the enormous honour of being the first to use this auditorium, which has blackboard and chalk and all the technology we need. Doesn't have a clock, so I could go on. Eventually, maybe the society could sponsor a sorry, yeah, clock. 20 minutes, isn't it? Yeah. Okay, I'll try and keep time.
Nick, is your mic on?
Is the mic on? Don? No one can hear me. Yeah. Hey, can you hear me better? No, no one can hear me. Can't get it much closer. Swallow it.
It's fine. It's fine. You've got it.
It's on.
Yeah, you've got it.
Yeah. You just have to pay attention.
You may
Ah. Okay. Okay, good. And it's tricky because I'm facing this magnificent view, so I get distracted, but like you're looking at me, so you have to pay attention. Right. Okay, so modeling complex traits. What do I mean by that? Where are my arrows? Here we are. So, I mean, a complex trait is really any trait that has an unclear genetic basis involving a few loci, many loci. We don't necessarily know, usually we don't. And so it covers, a huge range of traits. In fact, most of the things we care about with disease risk, crop yields and so on, one can see are complex traits. So what I'll be doing is contrasting two very different modeling frameworks, modeling traditions. One, quantitative genetics, where we have a more or less phenotypic model, which is quite a straightforward model, as I'll try and convince you, which works pretty well, but doesn't say anything about individual genes. And then on the other hand, we have population genetics, which tries to construct very detailed models based on the properties and the effects of particular genes. So I'm talking about the contrast between these modeling approaches, but at the end, very briefly, sort of try and make some more general points about our approach to modeling. To what extent do we need to have a microscopic model that justifies our broad brush phenotypic approximations? And maybe to get a sense of it, how many people are kind of familiar with population genetics as opposed to all the other fields we cover? Put your hands up. There's too many of you. Okay, so probably it's interesting. I always think of us as a small, quiet, obscure field, and yet, more than half the audience. What about epidemiology and ecology and phylogenetics? Yeah, or 1, 2 hand. Okay. Oh. Yeah, it's an interesting bias. I don't know where it comes from. Anyway, okay. So complex traits. I mean, a long-standing observation going back to Darwin or before is that, artificial selection works. That's why we're on the whole well-fed, and, why there've been such enormous, or at least part of the reason why there've been enormous advances in plant and animal breeding. So if you select on more or less any trait in more or less any outcrossing sexual organism, you'll get a change which over tens of generations changes the population over many standard deviations well beyond its original range. And actually in long-term experiments in reasonable sized populations, this response keeps going until we kind of lose energy or grant funding, for 50, 100 generations, right? And this really immediately implies that heritable variation has to be typically polygenic. If there was just one or a few variants that you could pick out and fix, that would be the end of it. Things keep going, and that implies there are a lot of variants of small effect. We also know, it's a slightly more indirect argument, that at least for the first 50 or so generations, this is not new mutations. This is variation in the population. Okay. So recently, this has been confirmed dramatically, I think, beyond most people's expectations. It's not just that variation in complex traits is polygenic, it's based on very, very large numbers of variants. So the classic example is human height, which is, of the order of 70, 80% heritable. And there have been enormous human genetic studies, not because people are interested in height so much as using it as a model for understanding disease risk, etc. And I think now actually even more of the variance in height has been assigned to particular genetic variants, single nucleotide polymorphisms. And this study I've quoted, I think even this is a bit out of date now, found that most of the variants could be attributed to at least 12,000 variants across at least 21% of the genome. So there's enormous numbers of variants, which means, of course, that we can't really profitably study those variants and their interactions directly. And so we come to a model that's known traditionally as the infinitesimal model, which waves away all of these genetic details. Yeah, don't show it again. So the infinitesimal model is rather amusing or even disconcerting that this is known as Fisher's infinitesimal model and attributed to some section in Fisher's 1918 paper on the correlation between relatives on the assumption of Mendelian inheritance, this classic paper. Somewhere in there, he proposed this model. He never called it the infinitesimal model, and it's not completely clear exactly what the relation is, but it came to be called the infinitesimal model. Papers on the infinitesimal model started to be published in the sort of 60s, 70s. But if you asked people like Michael Bulmer and Bill Hill, where the name came from, they said, "Oh, I can't remember." So it's a very obscure thing, and it's not really been defined precisely until Alison Etheridge and Dean Weber and I sort of did a review for which ended up in TPB, even though it was kind of well understood and used and was the practical basis for most quantitative genetics. And the model is extremely simple. We suppose, as the usual observation, that we have a trait that is normally distributed, and you take two individuals from that trait. Let's just think about the genetic component of the trait, which we can define, the breeding value. Then the distribution of the breeding values in the offspring is also a normal distribution with a mean at the average of the two parents. That seems obvious that it should be. And with a variance that is independent of the values of the parents. So that's the crucial assumption in the model, and it's empirically works rather well. That there's a certain variance, called it V0, which is independent of the parental trait values. And that means that if you select on the population, select the big ones or the small ones or whatever, you will not alter the variance. And that is a dramatic simplification. And there's a slight twist to this, which is that the variance amongst offspring isn't entirely constant. It does depend on the relatedness between the parents. So this variability in the offspring, the variation in genetic value of the offspring, is generated by recombination, by the mixing, the shuffling of the genes that come from the two parents. It's the shuffling that occurs in the meiosis that generates the sperm and the egg that produce the offspring. And so clearly, if the two genomes in either of the parents are identical to each other, if the parents are inbred, then that shuffling will generate less variance. And we can quantify that by saying that the variance amongst offspring is V0 times 1 minus F, where F is the average identity by descent between the genes that come together in meiosis. And that's something that sort of, I've mentioned genes, but really it's something you calculate from the pedigree. If we assume some distant population that was a set of unrelated founders, and we know the pedigree, then we can work out what's the chance that these two genes will be identical by descent from the same gene in the ancestral population. And to the extent that they are, that reduces the variability. So variability does change, but due to inbreeding, due to something that we can calculate from the pedigree without saying anything about selection or trait values. So just a little bit of history, and this goes back to ideas of blending inheritance, which was really the first, or amongst the first quantitative studies in population genetics, I suppose, even before genetics. Fleming-Jenkin had criticized Darwin's theory of evolution by natural selection by saying, well, if you take two parents and you produce the offspring with some mixture, some blending of the two parents, and you dissipate variance. And then Davis, and there was even some other papers in the 1870s, showed that you would actually reduce the variance amongst offspring by 50% per generation. So it's really very hard to see how you can maintain variation and drive natural selection. And Galton took up this problem and introduced a lot of, statistical measures such as regression, introduced a sort of large-scale collection of data on human height and all sorts of other rather strange phenotypes. And he observed that, offspring follow a Gaussian distribution which is independent of the parental traits. And there's a nice quote here from really quite a deep paper in 1877 in Nature. in which he reported, amongst other things, experiments in which he got his aristocratic friends to take packets of seeds. He measured the size of these seeds. These were sweet pea seeds, I think. And they crossed them and their gardeners grew them up. And then he collated the evidence on seed size amongst the offspring. And he found that he was certainly astonished to find the family variability of the produce of the little seeds to be equal to that of the big ones. But so it was, and I thankfully accept the fact, for if it had been otherwise, I cannot imagine from theoretical considerations how the problem could be solved. And what he meant there was that if there was some complicated relationship between the sizes of the seeds in the parents and the offspring, it would really be a hard problem to disentangle. But actually, he could just take that variance amongst offspring as fixed, and he actually did calculations on the equilibrium between stabilizing selection and recombination under the infinitesimal model, which were later done by Bulmer in the 70s, actually. So there was tucked away in the appendix, one of the first calculations of an equilibrium for the distribution of a quantitative trait. So that's a little bit of the history. How does one justify this model? Well, traditionally, the justification attributed to Passage in Fisher is that it's the limit of an additive model in which you just have a pile of different genes, unlinked genes, which each have a small effect. The effects add up. And if you have m loci and the effects are of order 1 over square root m, then the variance is independent of m. It's the sum of squares of the effects. And as you increase the number of loci, as you go to this infinitesimal limit, you get the same genetic variance, but you find that basically selection has hardly any effect on the frequencies of alleles. Okay, I'll come back to why that is in a minute. But basically, if you imagine selecting on a population, you will shift the frequencies of the alleles to the extent they increase the trait and you select for an increased value of the trait, you'll increase the frequency of those alleles, but the effect will be of order 1 over root m. Now, if you add up a lot of small perturbations and look at the effect on the mean, that is a substantial effect. So we can predict a substantial change in the mean, which is proportional to the total genetic variance we're keeping fixed. But the change in the genetic variance is of order 1 over root m. It's smaller, and therefore we can ignore the effects of selection on genetic variance, etc. So that's straightforward, except that proper biologists then scoff at this and say, obviously organisms are not additive, it's a stupid model, how can we possibly use this? Well, we say it works, kind of, but it's very implausible. And so one of the things we did in this 2017 paper was to actually extend the justification really by applying sort of different flavors of central limit theorem to show that if you have a population with a larger number of genes affecting a trait with not too much interaction. There are constraints on the amount of gene interaction you can have, but nevertheless, you can allow some degree of gene interaction. This is a bit of an unfinished project, but basically we're trying to expand the realm of justification of this infinitesimal model. Hang on time. Yeah. So yes, okay. Okay, so if you think about it, if you allow any kind of gene interaction, then the model must break down. You've got a normally distributed trait. Suppose I just say, let's take the trait squared or the trait cubed or any nonlinear transformation of the trait. That will clearly break the theory. It'll produce an arbitrary distribution, right? So we can't allow any old epistasis, but the basic constraint, I won't try and express the mathematical constraint, but it's to say that you can't have any particular interaction effect having too large effect on the trait. It's a bit of an extension of the additive model saying we have very large numbers of genes, none of which has individually a very big effect on the trait. Similarly, you have to have a similar sort of constraint on the effect of any one epistatic interaction and also on the degree of systematic effect. If all the epistatic interactions point in the same direction as they would if you did a scale transformation, then again, that breaks the model. But we can sort of set up formally a model where we have a trait, which is a sum of additive effects, that's middle term there, plus a sum of pairwise effects, which scale with the number of loci, so that each of those additive and pairwise components are similar, and we can sort of push through the justification of the infinitesimal in a similar way. What we have to do now is extend it by tracking for each individual its additive component and its pairwise epistatic component. So an individual now is characterized by a trait value, which is a non-genetic component plus an additive component plus a pairwise component, and so on. And we can still write down the ways in which those components vary. And in fact, the traditional classical quantitative genetic theory already told us how to follow the changes in additive and non-additive variance components under varying degrees of inbreeding. We have to grapple, which is rather painful, with four-way identities between genes. We don't say what's the chance that this gene and this gene are identical by descent. We need to know what's the chance that this gene and this gene are identical, and this gene and this gene, or the chance that all four are identical. We have a bit of a zoo of F coefficients. But in principle, you have the same sort of scheme in which you can argue that the variance components with epistatic genetic basis will vary, but due to inbreeding and not due to selection. Okay, so these are just some simulations showing how the additive and non-additive components of variance on the right, of the mean on the left, how they change under neutrality, the dotted line, with strong directional selection, solid lines. The top line on the right is the additive variance, bottom line, the non-additive. And they do diverge, but after about 30 or 40 generations. So in some rather unquantified sense, actually, sorry, 5 minutes. Yeah. The model, works for some number of generations, tens of generations. If you have interactions, it works for a little bit less time, but it works in the short term. And that's really the realm in which we've justified it. So I've been warned by the clock, so I shall just summarize that by saying that really what we're saying is not that we have to have an infinite number of strictly additive alleles, but we have to have a situation in which the variation of the trait, potentially due to all the genes milling around in the population, is very much greater in principle than we actually observe in the population. And we sort of see that under experiments on artificial selection. We select on the trait, it can go many standard deviations this way, many standard deviations that way. And that means that you're getting very little information from the particular trait value of two parents about the possibilities amongst the offspring, because the alleles in the parents are spanning, if you like, or the trait values in the parents are spanning a much smaller range than what is possible given the standing variation. And we kind of know that empirically anyway. So it's a rather, it's a general qualitative justification. And it's a big open question, I think, how far one can define what models satisfy this infinitesimal scheme, which we kind of use empirically so much. So I think I'll skip over this very quickly and just flash past to say that, we can ask the question, under this model, what limits selection? And this goes back to a nice paper by Alan Robertson in 1960, in which he showed that because the additive variance under this model decreases by 1 minus 1 over 2n due to genetic drift per generation, you can predict how much inbreeding reduces genetic variance. If you simply add that up over the generations, you say that if you select on a population, the change over the whole time span of selection until you've exhausted all the variance is just 2ne times the change in the first generation. And you can actually extend that to include epistasis with some slight changes. And Robertson also pointed out that you could derive this result by seeing that what's happening is that the fixation probability of plus alleles that you're selecting for is slightly perturbed under the infinitesimal model. And that what's really happening is that we're assuming that selection on each allele is weak. The real assumption, the real basis of the infinitesimal model is that n times s is small, such that the dynamics of any particular allele is largely neutral. So you're simply perturbing the neutral situation. The allele frequencies can change a lot, but if they change, it's because of drift and inbreeding, not because of selection. So to summarize all this, the infinitesimal model is used in practice as really the foundation of quantitative genetics. It explains the short-term evolution of complex traits, and it really fits quite well over sort of 50 generation timescales in smallish populations. Whether it applies over longer timescales, whether it's actually useful to us for evolutionary biology, that's another thing. We can justify it as the limit of an additive model, but it applies more widely. Clearly, it breaks down if there's linkage, and there is linkage. We can't have an infinite number of genes all on different chromosomes. And it's actually a bit of a puzzle why it actually seems to be rather robust to the actual limited genetic map that real organisms have. So finally, just to come back to the question I raised at the beginning, we have here a model which was was used and justified empirically, just seemed to work. It seemed to describe the statistical structure of inheritance. It came out of the biometric tradition around 1900, which tried to simply come up with a statistical explanation for quantitative traits. And yet, in contrast, we have this underlying complicated world of population genetics that at least half the audience loves, I hope, or hopefully. So, these seem very different flavors. One is a very simple model where you just follow the statistical properties of individuals and trait values. The other is a very detailed mechanistic basis. And one can take a certain class of very, very restricted population genetic additive models and justify the phenotypic approach. It's rather like what we do when we do population genetics, in which we assume the diffusion approximation, and that tells us how allele frequencies will change in the long term if selection is sufficiently weak, etc. We justify that using the Wright-Fisher model, but that again is a very specific model. Do we actually need to do that? Can we just live happily with the phenotypic level models? That's it.